Hey guys, welcome to The Kitchen Think, a place where we talk about events that are important to us and are also hopefully important to you. And today we're talking about vaccines against COVID-19 with a specific focus on the mRNA vaccine. Now, the reason why we chose this topic is there's mainly two reasons. Number one reason is that it's everywhere around us. Everybody's talking about it, social media, news, people are getting injections of it, and so
We're talking about it all
We're
the time.
talking about it in our kitchen where we are now, of course, and so on. Secondly, the reason why we chose this is because we know something about this topic. And the reason why we know something about this topic is because both myself and Veronica, we have PhDs in biology. And although we haven't been working in biology or in biological field for some time now, we have been in the past working in academic settings, in pharmaceutical industries for probably over a decade. So we do know how the scientific process is done. We do know something about biology. We do know about the research and so on. So let's kind of dive into the topic of mRNA vaccines. And the first thing we probably should talk about, Veronica, is people are afraid of this vaccine a little bit. And it's understandable, right? When you're talking about something like a new technology, such as something like an mRNA vaccine, which is a new technology, now you're taking this technology and injecting it into your body. And of course, now people are worried. Is it safe? Is it going to hurt me? Is it going to help me? And all these are very valid questions that we'll hopefully talk about here in this video. If not, we'll talk about in the future videos. But before we kind of address that, we should say that actually mRNA technology as therapeutics, not as a vaccine, but as therapeutics, something that's going to help you fight infections and diseases, the idea behind it has been around since 1990s. And
yeah, that's right. I think in 1992 or something like that, an experiment was done where mRNA was injected directly into granted mice, but to basically cure them and help them with their diabetes in a way. So granted, it's in mice, and ever since, obviously, the mRNA technology field has been evolving.
Yeah, that's right. So even like I said, from the 90s, there have been already experiments done in animals where they've showed that mRNA can be used as a therapeutic agent. Now, of course, it's a huge difference between injecting something like an mRNA into a mouse and now creating a vaccine that's injected into millions and millions of people. But first of all, almost 30 years has passed since. And secondly, there's been a huge spike in research lately around this topic of mRNA. And let's talk about that a little bit because it's actually quite an important topic that a lot of people overlook. Scientific research, like anything in life, is driven by money. And the more money is devoted to a specific topic, the more the topic progresses, right? Science is expensive. You got to pay your personnel. You got to pay reagents, lab space, equipment. It's actually extremely expensive. So usually priority as far as funding goes is given to topics that are of pressing nature. So meaning something like, let's say, breast cancer for a while, ovarian cancer was another big one, heart disease, anything that's of a pressing nature is usually given a lot of funding, and people focus on that to help society deal with those type of diseases. mRNA therapeutics, although it was interesting, it hasn't really been a pressing need to create any type of therapeutic agents associated with mRNA. So the progress between 1990, I should say, when the first injection of mRNA has been done into mouse, and now has been quite slow, but still some progress has been made. And now, all of a sudden, COVID came about. And COVID is a driving factor behind a huge progress in the scientific research associated with mRNA. Now, why is that? Well, the answer is very simple. As soon as COVID happened, people said we need vaccines, and we need them fast. Now, traditional vaccines take a long time to design. We'll probably talk about that later. mRNA vaccines, their huge advantage is the speed at which they can be implemented, at which they can be designed. So all of a sudden now, all this money from different nations has been given to all these different scientists who have spent significant amount of time working on mRNA technology, exponentially growing the scientific field behind it, cultivating in mRNA vaccines that we now have. So although, like we said, the technology is new, the topic has been brewing for a very long time and has cultivated into this vaccine that we now know. All right, so let's dive more into the mRNA vaccines, the mRNA technology, vaccines in general. And what do you want to start with, Veronica?
Yeah, so I think maybe before we go into mRNA vaccines themselves, we should talk about vaccines. And one of the first things that we like to do is to sort of define what a vaccine is. And from my personal perspective, a vaccine is a biological agent that you can use to basically teach your body how to defend itself from a foreign agent, like a virus. So in this case, SARS CoV-2, which is the virus that causes COVID-19. So that's sort of generally how I would define a vaccine. What about you, Yuri? How would you?
Yeah, it's a good way to boost your immunity. So basically, all the animals, and humans included, have a way to fight infections for the most part caused by small pathogens like virus and bacteria through immunity. So building antibodies, different processes that destroy the invading kind of organisms, if you will. And getting a vaccine basically boosts that immunity. It gets a head start on the process. So later, if in a later day where you were to encounter the real virus, you would deal with it much, much better, much sooner, and so on. So it's a great way to combat diseases in general. And actually, historically, vaccines have eradicated lots and lots of diseases around the world. So history is in our favor. And we're hoping that, in a way, this vaccine or this series of vaccines will also eradicate COVID-19 eventually, and we'll go back to our normal way of living.
Yeah, I think the next thing that we wanted to do is sort of talk a little bit about vaccines in general, and then dive into mRNA vaccines. So how vaccines are made. So we actually put together a few slides to sort of visually demonstrate or show to you how vaccines are built or developed.
And while Veronica is showing you all these figures and slides and talking you through the science behind it, I'll be manning the computer, manning the camera. And then once we're done with that, we'll meet here again and we'll say goodbye.
All right, so here's our first slide. So this slide, although it's a little bit complicated, we won't cover everything on the slide, but it's a way of graphically showing you how vaccines are made. So on the left-hand side of this slide, it's sort of a graphical representation of how traditional vaccines are made. Usually what people would do is take the virus and either completely inactivate its virus capability or partially inactivated, what we call an attenuated virus, and use those virus particles, if you will, and inject them into the body so that your immune response would create the right antibodies to basically fight any future infections by the real virus. Now, these actually take a really long time to engineer and design and grow in the matter of years, actually. What you can also do...
Let me stop you there for a second, Veronica. So with these two type of processes, these have been the traditional approaches to vaccine development. Is that correct?
Yeah, that's actually true. Yeah.
So the whole inactivated virus or attenuated virus, as you can see in figure A here, attenuated means partly deactivated. So it's still functional, but not as functional, it's not as infectious as the real virus. Both of them require the use of a true virus. So that means that laboratories have to be specially equipped to design these viruses, to purify these viruses, to work with these viruses, to deactivate them. Safety protocols have to be followed. It's a very long process and it works very, very well. But when time is of essence, as it is now with COVID-19, these, although are still very kind of good ways of creating vaccines, just take too long. Yeah. That's really the idea.
Yeah, and there's other sort of methods that we're not necessarily going to go into, but you can also consider actually taking peptides or pieces of proteins. They're part of the virus. So it's not the virus, it's just a little piece of the virus. And these peptides or proteins can be injected into the host, the human, and that can also create a immune response, meaning exactly what you want a vaccine to do. These are a little bit definitely easier to make. Even just the graphical representation is much more simple, but it takes still some time. The last couple of approaches to making vaccines are what we call nucleotide-based vaccines, DNA and RNA. Now, let's not talk necessarily about DNA right now. Let's just go directly to RNA, since this is the really topic of this video. So just before I go into more details about RNA, as you can see graphically how simple this RNA is, and we'll talk about what RNA does in the human body and how you can exploit what it does in the context of vaccine development. Anything else you want to add, Yuri?
So if I understand correctly, Veronica, so we talked about A and B, so the live and the whole inactivated viruses.
Yep.
We talked about the protein production, so point E,
yeah,
where basically you're just taking the protein that's part of the virus and injecting that into your body. Still takes a long time to purify and make. And then, of course, you have the nucleic acids, DNA and RNA, which we'll get to in a second.
Yeah.
point D is interesting. We're not going to talk about it, but I believe this is what AstraZeneca is using.
That's correct.
Basically, in a way, how do I explain it simply, is to take another type of virus that is less infectious, like a common cold virus, deactivate it, and insert into it parts of the COVID-19 virus and inject that.
Yeah.
And then that original virus replicates within your body, producing an immune response to COVID-19. It's also a very interesting technology. Maybe at some point we'll make a video about it, but at this point, let's forget about it. Let's not kind of overwhelm you. So let's go into RNA and talk about how that's simple and how can that be effective.
Yeah, so basically the next slide I want to talk about is actually RNA itself. So RNA, more specifically mRNA or messenger RNA, it's sort of a signal or a sequence that basically only happens inside of the cell. So if this mRNA is found outside of the cell, it actually gets degraded quite fast. It needs to be inside the cell to be relatively stable. And relatively stable, we're talking about hours to maybe a day, but it doesn't stay around for very long. What it does inside the cell, this is outside of the nucleus where all the genetic material is. What it does outside of the nucleus inside the cell, if it's recognized by this enzyme called a ribosome, the ribosome will read this information, and that information will sort of teach the cell how to make a protein. And then these proteins obviously can stay inside the cell or go to the surface of the cell or outside the cell, and then basically are components of the cells, the body, and that's sort of the idea. And this process by which an mRNA is turned into protein is called translation. One important thing to highlight yet again is that mRNAs are not stable at all. So if you inject it into humans, and we'll talk about it a little bit more later, they actually get degraded quite fast. But this is sort of what mRNAs do normally. Anything else you want to add here?
Yeah, so let's talk a little bit about this diagram, because maybe a lot of people are forgetting their basic high school biology here. So let's just remind you people of this, because it's actually important to talk about this. So what you're seeing down here, this is the nucleus. This is where your DNA is. Now, DNA basically is a code for all your proteins that basically drive your body in a sense. DNA itself cannot be read unless it's first converted to RNA. And this is the process. So DNA is converted to mRNA. This is called transcription, which is not shown here. And that mRNA now is read by the ribosomes, like Veronica explained, and is converted into a functional protein. Yeah. This is a natural process within your body. And the good thing about it, as Veronica said, and this is important later on for vaccine development, mRNA occurs naturally in your body. It doesn't have a long shelf life, so it's going to get degraded quite fast. So any excess mRNA that is not used, it gets destroyed by your body naturally, so less chances of side effects and stuff like that. And it never gets into your nucleus, so it can never get reintegrated with DNA again. Also very important for vaccines, because if you inject mRNA, the chances of it being somehow integrated into your DNA is almost zero. Probably is zero, actually. So this is an interesting process that's, kind of as a gateway to how vaccine based on mRNA technology works.
Yeah, and that's basically what we're going to be talking about next. So basically what scientists did was to take this mRNA that would produce, if read by a ribosome, a protein called a spike protein. So the spike protein, actually, you probably have seen this image quite often in the media or on the internet and social media. The spike protein is actually all around the surface of the SARS CoV-2 virus. So again, this is a virus that creates or causes COVID-19. So scientists took this mRNA sequence that we know would produce the spike protein of the virus. And the thought was, well, if we can produce the protein, can we actually use with that mRNA, can we actually inject that mRNA into humans and let the natural occurrence of this translation reaction occur in the cell of human beings? Why this? Because it's such a faster method to just make RNA and inject it in humans. Did you want to add something to this sort of image?
No, the image is great, and the explanation is great. But remember what we talked about in the beginning of this video, how this technology, the idea of using mRNA for therapeutic, has been around since the 90s. That's exactly what they did when they injected a mouse. Right. They took an mRNA of a specific protein, injected into a mouse, and that translation happened, creating created a specific protein that helped the mouse deal with diabetes.
Yeah.
So that idea already existed for a while. We're just taking it and converting it into more of a immunization type of specific technology, if you will.
So one of the advantages of mRNA as a therapy, if you will, is the fact that it's, like Yuri mentioned earlier, it's easily degraded. So it doesn't stay around for very long. So there's no worries about any sort of like safety issues because you have this mRNA hanging around. But it's also actually also one of the hurdles, scientific hurdles that mRNA vaccines had to go through. How can we control or how can we stabilize this mRNA long enough that we benefit from the immunization, let's say, of the process of the vaccine working? So that's what the sort of next diagram is showing. So what they did is they took these mRNA pieces and they sort of packaged them into what we call these lipid nanoparticles, or sort of basically fat blobs or vesicles or droplets, if you will. And these lipid droplets, which are full of mRNA, is what these mRNA vaccines like from Pfizer and Moderna are. So what the vaccine, the injectable vaccine is basically these fat droplets. Don't worry, it's not going to make you fat, but it is going to carry these mRNAs. Did you want to add anything else, Yuri, about sort of this?
Yeah, so Veronica mentioned a very good point. mRNA is not very stable, which is a good thing if you're injecting a vaccine based on mRNA. So any excess mRNA gets destroyed, but it's also a hurdle that you have to overcome to stabilize the mRNA during the delivery process. Yeah. That's exactly what they did here by kind of using the natural or mimicking the natural structure of your cell. If you go back to the previous slide for a second, Veronica.
Yeah, let me see if I can go back. Give me 1 second.
So you look at this slide. You see this outer membrane here? That's the cell membrane. It's basically done the same way. You have your hydrophilic area, which is because it likes water. And then you have your hydrophobic area, which is your lipids, your fat that's on the inside. And that's what keeps the cell intact. And what they did is basically mimicked this structure by creating this kind of almost like an artificial cell membrane that is encompassing the mRNA.
Yep.
And that really stabilized the RNA, allowing it to be injected. Now, I know also for a fact they also added some modifications to RNA to make it more stable, to mimic more natural mRNA that you have within your body, so the body doesn't see it as a foreign entity and says, oh yeah, this is just like any mRNA within my body, and deal with it in the same fashion.
Now, once the mRNA vaccine is injected into you, then what happens exactly? So this is what this next figure depicts. So Yuri actually perfectly led actually leads to the sort of this image. So this is this lipid particle that's sort of like surrounded by this sort of this what we call phospholipids, or fat droplets around, and the mRNA is inside. This sort of lipid, these lipids, this fat, really likes this other membrane that Yuri was referring, the membrane of the cell. And it likes it so much that they actually when they touch, they fuse. And when they fuse, they sort of like start joining together. And that opens up this fat droplet and delivers the mRNA inside the cell. We told you this is where the mRNA it's supposed to be. That's where it's most stable also. And once it makes it inside the cell, then if there's ribosomes around, the ribosomes will recognize the mRNA and will basically translate them into proteins. In this case, since the mRNA was built specifically to make the viral spike protein, then the spike proteins will be made and then will be basically exposed to the outside, to the body, and the body will recognize this as a foreign agent because it's basically a spike protein of the virus. And this will activate an immune response where antibodies will be made and T cells will be activated. That's sort of generally how it works. And eventually the mRNA will naturally be degraded anyway, so it won't stay around for a very long time. But we know from all the experiments that have been done over the years, we know that it's there long enough to be able to now create an immune response. And now your body is ready to fight the real virus if it infects you, because now the immune system will recognize these little spike proteins on the surface of the virus and knows exactly what to do because you taught it how to do it with the vaccine. That's sort of generally how it works.
Yeah, it's a really interesting idea, right?
Yeah.
You're basically using your natural body's ability to read this mRNA and convert it into proteins to introduce foreign mRNA and convert it to foreign proteins that are part of the SARS CoV-2 that are causing the immune response to SARS CoV-2. Now, I know a lot of people are going to ask, can this mRNA make me sick with COVID-19?
Right. So it cannot, because the mRNA, these pieces of information that are inserted to the cell, they actually only produce a very small part of the spike protein, and actually doesn't even produce the entire piece of the spike protein. It's just a tiny little piece. So it only produces this part of the virus. It does not or cannot, or this mRNAs are not able to recapitulate or rebuild the virus inside of the cell. That's what makes it so safe. mRNAs are naturally occurring in the cell. And we've made a very small part of the protein. So there's no virus in here. There's just a small piece of the virus that's sufficient to activate your immune response.
Yeah, it's really cool. So basically, you cannot get sick from it with COVID-19 because you're not inserting the whole virus, which is what the traditional vaccines always use. The whole virus is attenuated or deactivated. This is not using it, so there's less chance of you to get sick, pretty much zero chance. It doesn't require much time. It doesn't require much purification. It should be much safer in general. Yeah. Will you still get a little bit achy? Yeah, because immune response kicks in. So immune response kicks in, you get a little bit watery eyes, a little bit sniffly, potentially even sometimes a little bit of inflammation at this point of injection. That always will happen, but it's not that you're sick. It's just that the immune system is working. It's a good sign that it works well. So yeah, it's a really, really cool way of kind of introducing this new vaccine.
Honestly, when you first sort of look or understand how it works, and hopefully you guys now understand a little bit better, and feel free to ask us questions in the comment section. It actually is sort of an amazing technology, and you almost wonder how come this was never done before, because basically what it has done, the mRNA vaccine world, what it has done is shorten the length to a useful vaccine from years to literally months, right? I mean, we've this has been about almost 10 months into COVID-19 pandemic, and within that amount of time, it's unheard of. So to a lot of people, it's extremely surprising, and it's very scary that almost you think, how can they make a vaccine so fast? But hopefully by understanding how simple an mRNA is, how simple it is to make, and how much you can actually make of it, it will make you understand that this is sort of a very neat technology that was already there, but we decided to exploit it because we needed it. We needed it now. We needed a vaccine right away.
All right, guys, let's wrap it up. And I'm going to ask you, Veronica, a question that's probably on everybody's mind. Would you take this vaccine?
Yeah, I think at this point I will take it. I think the safety studies that have been done with even one of these mRNA vaccines had 40,000 patients in it, and the studies were followed for 2 months. Obviously now it's been longer since they did the studies, and I think almost 2 million people already received the vaccine just in the US. So I think at this point I would take it, and hopefully it will help us get back to normal life. What about you?
I would take it too. I mean, considering the science behind it, it's a vaccine. Well, first of all, like Veronica said, millions of people already have been administered this vaccine. They seem to be doing fine. Yes, there were some people that had allergic reactions here and there, but this is expected of vaccines in general. So I'm not too worried about that, especially now that we know it may happen from time to time. Having said that, again, this vaccine is super simple in the sense that we're not dealing with a real virus. So there's less chance of things to go wrong. Like, for example, imagine a virus that's been deactivated hasn't been deactivated properly. And now you got an injection, now you got sick. This eliminates that. There's no virus. It's a very simple process. I think it's quite safe. I would definitely take it. Cool. All right. Well, that's it, guys. So hopefully you like this video. Hopefully you learned something new. By the way, also tell us, would you take it or not? Tell us in the comments. We'd love to hear from you guys. But if you really enjoyed this video, give us a thumbs up, hit that like button, share it with your friends. If you just didn't like this video, give us a dislike as well. We'll take it. Again, leave comments. We'll be happy to answer your questions. Hopefully more videos will come in the future. Yeah.
Hopefully some of the comments that you put underneath this video will sort of lead to the next topics of the next videos. So see you in the next video.
All right, guys. Bye.